Hello, AP Bio folks. If you remember, last Friday we left off learning about protein folding and how a string of amino acids twists and folds. First, the backbones kind of connect with hydrogen bonds, and then the R groups of the amino acids interact to fold the protein into a unique three-dimensional shape. Today, we're going to continue with that idea of talking about the importance of the shape of a protein. So we're going to start with a quick example. So that example kind of demonstrated why the shape of a protein can be so important and what can happen when the shape is incorrect. The shape of a protein determines its function, and later this week we'll specifically look at that idea with enzymes, but it's true for all proteins. If a protein is misshapen, it can cause diseases. You may have heard of sickle cell anemia. You may have heard of mad cow disease, CWD. If you are a deer hunter, you've heard of that one. That's chronic wasting disease, which has been showing up more and more in the deer population in Minnesota. The kind of human version of this type of disease is called Creutzfeldt-Jakob's disease. So there are many, many of these diseases that have been linked to these prions that are misshapen proteins. So today we're going to talk about how a protein can become misshapen. Some of these things, like sickle cell anemia, you might be aware, is caused by a genetic mutation. And so a mutation alters the genetic code, the instructions for how to build the protein, which changes the primary structure of the protein. If you remember, the primary structure is the order of the amino acids, so the sequence that they get put in there in the first place is different. This isn't really the type of change of shape that we're talking about today. Mutations can change the shape of a protein, but really we're focusing more right now on that process of folding a protein and how its shape can be changed during that process. So we're going to be looking more at how environmental changes can disrupt the formation of the bonds that create the secondary and tertiary structures. One of the environmental changes that we're going to talk about first today is the idea of an acidic or basic environment, the idea of pH. Some of this might be review, but it's important to understand this idea and how it affects the shape of a protein. So this diagram kind of shows a reaction that happens between water molecules. Occasionally, when there's a hydrogen bond formed between two water molecules, one of the hydrogens will actually break off of one and attach to the other molecule temporarily. That creates what you see here as a hydronium ion. We more often just write this, it's kind of traditionally just written as an OH, that's not right. It's more traditionally written as just the H plus, since the rest of that molecule is just the water, the H2O. So we normally represent that, and that's kind of common almost everywhere that you'll read, that this hydronium ion is just written as a hydrogen ion. Since the remainder of this, the other H2O, is the water molecule, it's really that additional hydrogen that makes it behave strangely. The other one that's left is that hydroxide ion that has a negative charge. And so when we look at acids and bases, what we're really looking at here is the balance between these two. And this is happening constantly. These hydrogens kind of shift over and then come back. It's a reversible reaction, and it kind of reaches a balance. There's no net overall change, but it's constantly happening within water. These are really small amounts of hydrogen and hydroxide ions, but both of them are highly reactive. And so they have a big effect on the other molecules that exist within living things. In pure water, the hydrogen and hydroxide concentrations are going to be the same because you have the same number of water molecules, and those hydrogens are just kind of shifting back and forth. Substances that are considered acids are ones that can add additional hydrogen ions only. So that increases the concentration. Remember, these little brackets mean concentration of hydrogen ions. Bases are either substances that soak up hydrogens, which leaves a higher concentration of hydroxides, or they're substances that create additional hydroxide ions. In either way, the net result is you end up with more hydroxide ions than you have hydrogen ions. So those are the things that can change the pH of a substance, are substances that act as an acid or a base. And we measure this using the pH scale. There's a little bit of math listed on your equation sheet related to pH, but this is one of those where you will not be asked to calculate this. It's just something you need to conceptually kind of understand what pH is measuring. So the way that this scale is set up, do not tell me who came up with this, but the way this is set up, the concentration of hydrogen times hydroxide ions equals 10 to the negative 14th at 25 degrees Celsius. And so we can use that equation, if we know the concentration of one, to determine the concentration of the other. The pH measurement itself is the negative log of the hydrogen ion concentration. So it's really measuring more the amount of hydrogen that's there. So as pH decreases, then the hydrogen ions increase. So you can see the scale here kind of ranges from 1 through 14. A lower pH means it's more acidic, which means there's more hydrogen ions, those H+, than there are hydroxides at that end of the scale. The other end then has an increased concentration of hydroxides relative to the hydrogens. So we can calculate both of those numbers if we wanted to using this equation. And so it is important to know this pH scale to know that when something is more acidic, that means it has a higher concentration of hydrogen ions. And when it becomes more alkaline or more basic, those are the higher numbers on the scale. And that means it has more hydroxide ions. So make sure that you understand those things about pH. Because that's what will help you figure out how these ions interact with the molecules like the proteins that we're talking about today. Within living things, we need to maintain a pH pretty close to neutral most of the time. And we do that using something called buffers. And so I'm going to show you a clip of a video. This video was actually recorded by Mr. Olson about a week or two ago for our general bio classes. But he's going to show you a little demonstration with a substance called milk of magnesia that acts as a really good buffer. So we're going to take just 2 minutes or so and watch Mr. Olson's demonstration here.
Welcome back. We're going to talk about milk of magnesia. So milk of magnesia is a medication you can buy from the store. Okay, this is like the knockoff brand of it, but it really is just a laxative. Okay, milk of magnesia. So what I'm going to do is I'm going to grab some of this universal indicator. Grab some of this universal indicator. Put a couple drops in there. I'm going to shake it up. Okay. Now I got a couple more drops, guys, just so you can see it a little bit clearer. There we go. Out the way. Shake it up. Okay. And it turns pretty blue or purple. Okay. Proving that's a basic solution. Now, milk of magnesia, guys, is a buffer. Today, you're going to be learning about what a buffer is, and this is going to kind of help tie this in. Okay. So right now it's purple. I'm going to grab some vinegar, which I know is acidic. Okay. I'm going to grab this vinegar. I'm going to add a couple of drops. 1, 2. I'm going to stir it up. You guys see it change colors? Changes right back. So, let's do it again. Boom, boom, boom. Shake it up. Turns yellow, turns green, then back to blue. Okay. So I'm adding something acidic. And that buffer knows that. Okay. So it knows that this is basic. I'm adding something acidic. So it turns partially acidic, but it's going to buffer and it's going to go back to basic, try to maintain homeostasis. Okay. So I'm going to do this one more time. A couple things there. Keep adding, keep adding. You see it turns orange, turns red. I shake it up, turns green, it turns back to blue. Okay. Now, this is achieving homeostasis. It is staying here. The buffer is doing its job. But this isn't always going to be true. There is a point where I can overload or I can put too much acid in there where we cannot return back to a basic solution. So let's see if we can reach there. I'm going to just keep dropping pipettes in there. Shake it up. Turns to green, turns back to blue. Add some more. Shake it up. Green back to blue. Okay. Keep adding. Shaking. Oh, it keeps going back, but it's getting a little bit longer. Add. Shake. Okay, it keeps going. Add. Shake. It's getting closer. Keep going. again. Now, as you guys probably learned today, buffers and homeostasis go hand in hand. Okay, our body is a buffer what we're putting into our body, so we can actually maintain a stable pH, which in realistic means that we maintain a stable internal environment, which means homeostasis. Okay, if every time I added something that was acidic to my body and my body needed to stay basic, our body wouldn't be able to regulate it. But with things like buffers, we're able to help regulate it. Okay, as you can see, I put a lot of acid in here. I'm going to keep going because eventually I'm going to overload it. We're getting closer. Is it going to stay? Is it going to stay? Or is it going to go back? Keep shaking, keep shaking. Okay, it's really close now. Okay, it's pretty much almost at neutrals. Still a little bit basic. Take a couple more. Mix it up, and bam. Okay, this is now an acidic solution. I overloaded my milk and magnesia. I overloaded my buffer, which is a strong basic, and turned this into an acidic solution. Okay, this worked for a long time to help maintain homeostasis in the body.
All right. So a buffer is a substance that minimizes the change in hydrogen or hydroxide concentration in solutions. And as Mr. Olson explained, that's really important in keeping living things at homeostasis. Most living things need to be pretty close to neutral, with exceptions for things like the acid in your stomach, which actually has a pH closer to about 2 or 3. But that's only for that one organ to help it perform its function of breaking down your food. The rest of your body needs to stay pretty close to neutral in order to function. And so buffers work kind of like you see in this diagram here. Oh, here's one more example of kind of how buffers work. If you add 0.01 moles of a strong acid to water, the pH drops from 7 all the way down to 2, which is a huge change. This is a logarithmic scale. So remember, a change of 1 going from 7 to 6 is a change of like 10 times, 6 to 5. Now you've gone like 100 times. So going from 7 to 2 is a humongous change. But if you add that strong acid to blood, it only changes from 7.4 to 7.3 because of the buffers that we have. And the chemical equation that you see here gives you an example of kind of how those buffers work. This is showing how this molecule, H2CO3, acts as a buffer, and it can exist in these two forms, either as H2CO3, which is neutral, or as HCO3 minus, which is its ionized form, and there's that extra hydrogen ion. So you can see here, if the pH drops, you can kind of understand how this equation works. If the pH drops, first of all, if the pH number goes down from 7 to 6 to 5, remember what that means is we're adding more hydrogen ions. So if you increase the concentration of these, that's going to cause this reaction to move in this direction, because you have more of this reactant. So the hydrogen ions are going to end up combining with this HCO3 minus, and that will end up increasing the concentration of this molecule. If, however, the opposite happens, if there's a rise in pH, remember, if the numbers start going up 7 to 8 to 9, that means we're actually getting more hydroxides. So that's what a rise in pH means. Then one of the hydrogen ions from here, one of these hydrogens is actually going to break off and join with this to make more water molecules to kind of neutralize those hydroxides, which will increase the concentration of this. So this molecule will move in whichever direction is necessary in this reaction to help maintain homeostasis and keep the pH as close to neutral as possible. pH can have a big effect on living things. Like we said, you need to maintain the correct pH within your body, but the pH of the environment around some living things can matter as well. That's really important when you're talking about plants and soil pH. You may have seen these flowers before. These are hydrangea flowers, and hydrangeas are actually a really cool example of how pH can affect living things. The exact same plant can produce blue flowers when the soil is a certain pH, and pink flowers if the pH of the soil changes. And so I'm going to link this video. It's about 5 or 6 minutes long. If you're interested in learning a little more about the chemistry of this and just seeing an example of how the soil chemistry can affect the color of these flowers. But now back to our main topic. The shape of a protein matters to allow it to function. And that shape can change based on the environment. So the environment can affect how the protein folds, not the order of amino acids. So the environment isn't going to change the primary structure, but the environment around a protein can change its secondary and tertiary structures that cause it to have its specific shape. Some of those environmental factors do include changes in pH, which we talked about. We'll come back to that in a second. Things like salt concentration or temperature or other types of conditions. If the environment is changed, some of those weak bonds that hold the protein together might be destroyed. And we'll talk specifically about how pH and temperature can cause that to happen in just a second. The protein then loses its shape, and that process is called denaturation, or we say the protein has become denatured, which causes it to be biologically inactive. It can no longer perform its function, and you can kind of see that if this is our normal protein shape. When it becomes denatured, it kind of loosens up and loses that folded three-dimensional shape or conformation that it has. Sometimes this is a reversible process, depending on the environmental factors that caused it to lose its shape and kind of how completely it has lost its shape. Sometimes, if the environment changes back, the protein can fold right back up the way it was. Not always, but sometimes that's true. So how does pH specifically cause a protein to do this, cause a protein to denature? In the diagram here, you can see some examples of the R groups of some of the amino acids. You can also take a look, I believe it's page 57, that has all the different R groups and their chemical properties. So look at those and also think about the chemical changes that are happening when pH changes. And see if you can guess what types of interactions between these R groups could be disrupted by changes in pH. You can look back to the notes from Friday as well, and what level of structure we're talking about here. So one of the things that can be influenced by the change in pH is hydrogen bonding. And remember, hydrogen bonds happen because of polar covalent bonds within the molecule. So an example of this in the diagram is right here. We have this oxygen-hydrogen, this hydroxide group, which is a polar covalent bond. So the hydrogen end of this is slightly positive. And we have this carbon-oxygen bond here. And oxygen is more electronegative, so the oxygen end here is slightly negative. And that's why they attract each other and they form a hydrogen bond right through here. But if you add a bunch more hydrogen ions in here, they will be attracted to this negative as well. They'll be repelled by this positively charged end here. And the opposite, of course, would be true of hydroxide ions. So interacting with the ions that are in the environment around them can disrupt the formation of these hydrogen bonds and basically break apart those amino acids and start losing that part of the chemical structure. So that's one way. And hydrogen bonds, of course, are part of both the secondary and the tertiary structure of a protein. The secondary structure involves hydrogen bonds between the backbone of the protein. That's not shown in this picture, but it is part of the structure of that protein. And there are also hydrogen bonds between the R groups, like the one we just looked at, that are part of the tertiary structure. The other type of bond that can be influenced by a change in pH are the ionic interactions that happen between the R groups. So here we're going to look at an example. There's one of them over here up on the left, and the same bond is formed over here on the right. Those are examples of ionic bonds that form between the R groups of these amino acids. So the examples you see here are this amino group, that's that NH3 functional group. That's what the amino group looks like in its ionized form. It has an extra hydrogen, so it has a positive charge. And this one is a carboxyl group, that's COOH, but when it loses that hydrogen, it becomes a negatively charged ion. And so these two attract and form an ionic bond. But if the environment becomes basic, which means, again, that there are more hydroxides in the environment, then that NH3 is going to lose one of those hydrogens and become NH2, which is no longer charged, so it won't form an ionic bond. The opposite would happen in an acidic environment. That COO minus will actually attract one of those hydrogen ions from the acidic environment and become COOH, which again is neutral, and it's not going to form an ionic bond. So in any environment where the pH changes too drastically, these ionic interactions start to come undone, and the protein is no longer held together in those particular places. We're also going to talk just briefly about how a change in temperature can cause protein to denature. And to understand this one, you kind of have to think about what happens to molecules when temperature goes up. We're going to watch a quick animation to show you kind of what this looks like. So this is showing a protein in its three-dimensional conformation. You can see some alpha helixes in there, and it's all folded on top of itself. And you can see it's starting to move faster and faster. As things heat up, the molecules move faster, which eventually breaks the hydrogen bonds holding it together. And you can see that whole protein just kind of came apart. So as those molecules move faster, remember, hydrogen bonds are not super strong. And so those hydrogen bonds especially, it's kind of like if you were trying to hold hands with somebody while you were both running, and you're running at slightly different speeds and in slightly different directions, it would get increasingly harder to actually hold their hand, and eventually it would come apart. The same thing happens here. This is actually why it's so dangerous for human beings to run a really high fever. That really high temperature causes some of the proteins in your cells to start to denature and lose their structure, and then they can no longer perform their function. So the environment is really important to allowing proteins to maintain their shape so that they can correctly perform their function in your body. So here we are for the end of today. The information that we've been learning Friday and today about proteins and protein structure is section 3.5 in your textbook. And the part on pH is part of chapter 2, I believe. It's pages 37 through 39. So if you want to look back in the textbook to review, you can do that. Tomorrow, you do not have school. There will be no lesson posted. There's no assessment, nothing. The couple things that I would like you to continue working on over these next couple days is definitely study your functional groups. That quiz is coming up. Read the post on Google Classroom today to see exactly when your quiz will be. And we are also going to start talking about enzymes. So if you would, please spend a little bit of time over the next couple days reviewing the function of enzymes and the basic vocabulary that goes along with them. I will attach a Bozeman video to today's post that you could watch to help you kind of review that and get a little refresher on enzymes. It's about 11 minutes long. Or you can read through section 6.4 in your textbook. So enjoy your day off tomorrow. Quiz yourself on those functional groups, and I will see you in a couple days.